Hello and good afternoon. Welcome to this webinar organized today by Basque Culinary Center. We are broadcasting live from San Sebastian in Spain. We are truly excited to see such a diverse audience joining us from all over the world. A warm welcome to everyone attending this session. Today's webinar is titled Fermentation in Gastronomy, Fundamentals, Applications, and Career Opportunities. First of all, we will watch an introductory video about the master's degree in food fermentation, which will start this October at Basque Culinary Center's new location, GOE, Gastronomy Open Ecosystem. After that, I will introduce you to Cipriano Carrero, coordinator of the Masters. Cipriano will be in charge of introducing each of our 4 speakers today, one by one. Joining us are Diego Prado, Arantza Begueria, Francisco Kuhar, and Pablo Sánchez Rey. All of them outstanding professionals in the field of fermentation. And now we are going to start by watching the video.
You know that the groundbreaking of this master is that we put at the center of the fermentation, the author of the fermentation are the enzymes. Master in food fermentation is the first master around the world that is going to study fermentation from a gastronomic science point of view. First of all, we are having the social science. In social science, you are going to study how different cultures around the world using the art of fermentation. The second foundation is the food technology. In this module, you will study enzymology, microbiology, also biotechnology, and you will get like different tools to get data and to make decisions with all these results that you are going to get. And finally, the foundation of the culinary. Within this module, you will study the different ways that you can produce food, like a culinary point of view, like for a restaurant or in the industry. What kind of careers are you going to be able to perform when you finish this master? So you're going to be a food product developer that are going to be able to work for hospitality, restaurants. You will have all the tools for prototypation, or for industry. Also, you will have the scaling up tools. The possibilities of fermentation are infinite. Let's make the gastronomy wide.
Okay, so now I would like to introduce Cipriano Carrero, coordinator of the Masters in Food Fermentation. Hello, Cipri.
Hello, nice to meet you. Welcome everyone to this session. We are planning to have another one in July. But in this session, I want to talk about fermentation from the gastronomic science point of view. Fermentation, for us, is a collaborative act. We have different actors. We have microorganisms, could be bacteria, filamentous fungi, yeast. They provide like different enzymes, and to find the raw material is the second actor that we are having in the fermentation. Thinking about seaweeds, plants, vegetables, they give us the biomass, the different carbohydrates, protein, or lipids that are going to be being broken down by the enzymes or the enzymes coming from the microbes. And finally, of course, the humans as the directors of, or they feel that they are the directors of this orchestra, the culture and the technology. So what has left this collaboration along the history? They have left a delicious mark. Fermentation, we are going to establish just the basic. Fermentation, if you look for any book of pure science, it just, you read the definition, is a metabolic process that happens there are no access to oxygen. A lot of microbes have these metabolic routes, and it's a way that they are going to get energy breaking down different organic compounds, like more famous, for example, like sugar. And in this breaking down, they are going to produce byproducts for them, but for us, they are going to be ethanol, lactic acid, carbon dioxide molecules that are going to improve the organoleptic component, the texture of our aroma of our final product. For sure, it's familiar, this video that I show to you here. You don't know it, it's like it's a garum. It's a garum. We are speeding up the camera. You are not familiar with this. To produce this garum, here in BCC, we just took different fishes. You have here, for example, the salmonete, and we put it on this container with salt. Okay. Here, this bubbling that you are seeing happening, it could be happening from different lactoactive bacteria or yeast that are fermenting it. But the main actors are the enzymes contained on the stomach of these fishes. So if we look to the definition of before of the pure science, this is not fermentation. But you ask after, like, for example, to Diego Prado, that is going to speak later, he will tell you that he don't have any kind of doubt that this is a fermentation. So we are in gastronomy science. Fermentations within gastronomy science go beyond microorganisms. The principal actors are the enzymes coming from the raw materials or the microbes. So fermentation is not only that, because we are in a multidisciplinary discipline. Fermentation is a cultural, a technical, and biological practice woven together. So in this master in food fermentation, we have 4 principal pillars, which are first of all, the culinary, the social science, and the scientific. The next speaker is Diego Prado. I want to introduce to you. He is a research chef, having head of research and development of alchemists, having also coordinator of the Culinary Lab in Basque Culinary Center, and also having working in different restaurants as Faviken, Nordic Food Lab, or Noma. Diego also is going to be within our master in food fermentation, coordinating the culinary block, the module. So I would like you to invite Diego.
Hello, you hear me?
Yes.
Okay, so I was Cipriano was saying I'm a chef and a researcher, and I will try to briefly explain like what that means in the field of fermentation. No, I always use this kind of a title for some of my presentations, like how creativity, how we can really approach creativity and innovation in gastronomy, in the culinary part, through microorganisms and of course like through enzymes. No, so as Cipriano was saying, just a little bit about me. I have been working in many famous restaurants, but the main key is something like I would say very relevant. It's like I would say I was thinking of this just before this seminar. Probably 70 to 80% of all the projects I have been doing in the last 8 years has been about fermentation. I mean, fermentation has been like a real core technique. I mean, of course, fermentation is many millions of different techniques and a lot more science behind it, but it has been like really like a building block on where we have been constructing different projects, like many projects. So I will just like to show you. I would like select 3 projects that are completely different, that are all based on fermentation. And you will see like maybe, I mean, I guess if you are interested in this, on the fermentation, there are things that you probably have heard before, you have seen before, but of course it's like a little bit of a mindset of how we how we have been approaching fermentation to really use it, like I say here, like a starting point to do or product development or like dishes in like fine dining restaurants. No, again, you always try to, in all my career, you will see in this project, we are always trying to do, of course, new dishes that we create like these building block techniques through fermentation, but also pushing a lot to write scientific articles. No, so for example, this one, first one is something I guess absolutely everyone knows a lot about kombucha, but for example, this was actually a project that I did when I was a part of the Culinary lab in the Basque Culinary Center that we did together with Mugaritz, no, where it's a fine dining restaurant in San Sebastian, and the main key thing was to understand kombucha again is in a different way. Normally what you will do with the kombucha is you drink the liquid. So you drink this fizzy, soury liquid, but all the film on top that is actually like a cellulose film is normally always discarded. Taking some inspiration from fermentation from the Philippines that is called nata de coco, or that is basically same type of fermentation as kombucha, but the context is completely different because in nata de coco, you throw away the liquid, you don't use the liquid, but you eat the cellulose film. So we start doing many different trials with this. We develop like many different dishes from the restaurant, and you will see this and we end doing like kombucha films, the cellulose film from 1 day, from 5 days, from 10 days, 15 days, 20 days. But then one part again is you can do all these more creative type of dishes for a fine dining restaurant like Mugaritz, but we also push to publish this article. No, in this sense, this article is basically like a characterization and a protocol not say to teach, but to show to the gastronomic community how anyone can do this. That how how people can really understand and do all of this. So again, one of these essentially like how fermentation is one of these building blocks. Then, for example, something completely different. I've been working a lot lately with about insects and ants, but also directly related with the fermentations. For example, this one is something this is a forest in Copenhagen. When I was working before, we were collecting this type of ants that is called.
I think you are showing something. We don't see the screen, sir.
You don't see the screen?
No.
I'm sorry.
I do see the ants. The ants.
You can see?
Yeah.
Okay. Sorry, working. Sorry.
Cipriano's problem, okay? So I was saying, like, of course, working a lot with ants, but not only with the ants just for the sake of using the insect, but using the technological properties of the ants, And one thing we encounter with this, and I will, of course, this is directly linked with fermentation and also with anthropology. In this sense, you will see that in Copenhagen, in the Nordic countries, using ants is actually quite a common ingredient. I mean, you can just buy them everywhere. It's a not so strange ingredient to use. But what we're trying to do again is to use the technological properties of the ants and coagulate milks with them. So this is like some photos from my iPhone, like the different types of cream, different types of milks, like even some vegetable beverage like oat milk or almond milk, and how the actual ant has a crucial role in the fermentation part. And of course, we were saying about of course, the ants have some formic acid, and the formic acid drops the pH and all this. It was like a little bit more. We had like a very basic thought, but then at the end we realized that it was way more complex than that. That you can, and this is one of the ways you can coagulate depending on the temperature, the amount of the ratio, ants milk. Of course, the coagulation is different, but then we end doing this. And this paper. That again, one part was we did some dishes from the restaurant that we can show you some data, but then we add a lot of people to start doing this scientific paper. Veronica, for example, one ant expert, together with Rob Dunn, that we were doing all this, like the whole, I would say, entomology perspective of this. But one of the key things, I think, apart from that we have DTU Biosustain, Copenhagen University involved, was also, of course, Alchemist when I was at Alchemist, but also there's one person here, it's called Sevgi, that here is a Bulgarian professor that she has been researching all her life about yogurt and the origins of yogurt. And she found that the actual initially lactic bacteria from yogurt, that is something that is related, that is common in Turkey and Bulgaria. These actual lactic bacteria come from ants. So the oldest finding of lactic acid bacteria in order to do yogurt comes from these ants. No, and of course, when we start doing this, we have no idea about this. It was something that comes later with all of these, all the co-authors that are here are many co-authors with very, very different, how say, like specialities, very different specialty, but again, with one core thing together is fermentation. No? This is, of course, maybe a little more out of the box type of project, but still, like, it's very related with anthropology and the history of yogurt and everything. And one of the things we found just for this is like the thing ant has the 3 ways you can actually coagulate milk. You know, you can coagulate milk by just pH, that you add vinegar or whatever acid and you drop it to 4.6 and you coagulate milk. You can do with lactic bacteria, so you add any lactic bacteria, the lactic bacteria start producing lactic acid, you reduce the pH and you coagulate the milk. And then you can also do with enzymes that Cipriano loves so much, I would say. Like, of course, you have like things in Basque country like cuajada or mamia that you have rennet. That's normally chymosin. And also the ants inside has not enzymes that they use naturally to coagulate, that coagulates milk. So again, in one little pocket that is an ant, you have the 3 ways of fermenting coagulated milk at the same time. So it's very complex type of fermentation. And then we learn a lot. This is just some, we have a lot of data. But of course, something that I want to point here, for of course, we did spring and autumn, like seasonality. And of course, I mean, we understand like seasonality in many ingredients, but also the seasonality of ants, and also the seasonality, one is saying is ants, of course, but the seasonality of the microorganisms. If you can see, I don't know if, I hope you can see the screen. There's like a massive difference of the microorganisms you can find and the enzymes and the amount of enzymes you can find depending on the season. Also, that's of course for me as a chef is very like mind-blowing to think about like microorganisms and even ants as a seasonal product, no? So we did many different things, like from different ice creams, so like a kind of like a mascarpone, and also Esther did one of the like a meal wash technique, but using ants, no? And the last project I want to show that is also, I'm showing it because I think it's one of the most mind-blowing and more surprising, at least for me in the previous years, is about neurospora intermedia. Neurospora intermedia, this was a colleague, that a researcher is called Bayu. He's from Sweden, but he works at Stanford right now. And he actually he did like a little like a guest researcher trip to Basque Culinary Center when I was there, and then he did a research trip to Alchemist when I was there. And he teaches literally how to grow and show. And I have never seen this before. And I'm saying this that I have never seen this before because, of course, the beautiful thing of like this, the collaboration again through fermentation. So how someone like Bayu show us this microorganism that we have never heard in our lives before, and he teaches us how to do it. And of course, with all the knowledge that he has about everything, we literally learn how to, in a way, domesticate it. No, one of the beautiful things about the neurospora intermedia, as you can see, is the color. No, this orange color is like mind-blowing, and it really, for me, it really tastes kind of like mango, papaya. You know, when you grow like a good koji, it always reminds us like a pineapple or like very fruity. This is kind of the same, but like definitely for me, it's mango and papaya. And one of the things we learned, like it was very interesting, I will say it, is like when they grow normally neurospora intermedia this is called from Java, from Indonesia. They normally grow it in a very poor substrate, like so very low in nutrients. So when it grows very stressed, and it's always trying to survive. No, it's very, very, very stressed, trying to survive, and that becomes like very, very orange because it's always stressed. Because normally you grow it in okara, so it's the leftover soy pulp from soy milk, or the when you do peanut oil, all the press that is left. So it's very, again, poor substrate. And when we start growing it, we start growing in like rich substrate, like barley or rice or oats. And never turned orange. It was like very, very disappointing for us, but it never turned orange. So we learned with bio, of course, he teaches us that we can stress the microorganisms through UV lights. So we were growing in of course, like it's an incubation chambers, but we were putting cycles of UV light to stress the microorganisms and in a way to learn to do it like this. So this is the final dish we did at Alchemist. That is just a rice custard where we put the microorganism on top, like with this beautiful orange. They even look like Cheetos. That is very, for me, still very mind-blowing. But to do this, of course, was very a lot of time. It took us like a year and a half at least to be this consistent in growing neurospora intermedia. And also, all of this was because of bio and learning with scientists and chefs and to really take the best of both worlds. No? And then, of course, I was saying from all previous projects, we did this dish that is very minimalistic, but very technical fermentation-wise. But then, of course, bio, we did a few papers, one's a little bit more basic, like how to use it in culinary, and then a little bit more, I would say, complicated or more scientific in a way to analyze different things. And you see, of course, there's many people here. And we also did, we are also doing right now actually like a third article using the neurospora intermedia, but for doing liquids, like sake-ish like liquids. So saying that, just to finish and wrap up a little bit, is if you see, like you have three very, very different projects. The core thing is fermentation, and it has been, again, like the core building block on the techniques and everything in order to push forward in both fine dining, but also in like creativity and gastronomic research. That's it. Thank you very much.
Thank you so much, Diego. Very mind-blowing. Like a whole, like a difference, like a neurospora, ants, they are like animals. Kombucha, how we can get the different food application. Thank you so much, Diego, for to be here with us. We have to jump up in the next speaker. Is going to be Arantza Begueria. She is master in anthropology and ethnography, and she is one of the few researchers in Europe that are focusing, in the fermentation from the social side, anthropology point of view. She actually she's very active in different research projects. For example, I can say one here is lo que pasa fuera de los botes, pensar en la fermentación en Barcelona, but she is author of a lot of scientific papers, always with the anthropology and many of them studying the fermented food. So Arantza, I would like to invite you. She's also, sorry for not to say, she's going to be a professor in the Master of Food Fermentation. I would like you to invite you to.
Thank you, Cipriano. So, can you hear me?
Yes.
Okay. So, hello, everyone. Can you please share my presentation? There it is. Okay. So, thanks for being here and for inviting me. I'm going to talk about the social studies of the Master in Food Fermentation. So, I will talk about some of the issues that we will be discussing during the course. So, can you put the next slide? My name is Arantza Begueria, as Cipriano said. I'm an anthropologist specialized in food. I began my career working on topics related to health, food, and the body. And I have specialized in fermentation for the past years. I work now in Barcelona, where I'm researching fermentation practices of food activists, artists, lay people who ferment at home, and also fermentation professionals. So next slide, please. Next slide, please. Okay, thanks. So the French anthropologist Lévi-Strauss, which has nothing to do with the trousers, He once said that food is good to eat, but it's also good to think with. This is a very famous phrase of him. He meant that food is a great way to think about social and cultural issues. For example, food is a cultural expression of traditions, of religious beliefs, identities, be them individual identities or cultural identities or political identities. Food also marks social norms, taboos, moral values, and it's also a key element in our society because it's of its huge impact on economy, politics, and ecology. So next slide, please. In this master, we will learn that not only food, but fermentation is a fascinating concept to think about social, cultural, ecological, philosophical, political, or even creative elements of our society. We will learn about social theories on these issues, and we will think together about all of these elements that you can see on the slide. Next slide, please. So first of all, everyone talks about how fermentation is very healthy. Well, in this course, we're going to discuss the idea of health critically. So when we say healthy, whose health are we talking about? Our health, the planet's? Is this an anthropocentric point of view? So we are going to talk about what it means to be healthy, what it means to be natural, and what are the political implications of these notions. I'm not saying fermentation is not healthy, it is. But I think we have to think about this concept from a critical standpoint. So next slide, please. Fermentation is so fascinating that also proposes philosophical dilemmas. For example, if we are what we eat, you know the phrase, we are what we eat, and we eat fermented products with living microbes that are in us, are we still humans? Or are we microbes as well? Are we humans with microbes? Or humanhood is in fact made of many species? So fermentation gives a way to rethink what does it mean to be human in a context of a profound planetary crisis that we are living now. And it's also a very relevant question for the history of human philosophy, like what is humanhood? So we will be discussing about this. Next slide. We will also talk about the social dimensions of fermentation. Fermentation is a hub for social relationships, like a hub of social gathering, but also a place of social differentiation. So we will learn about how fermentation fosters social relationships through commensality, through culture sharing, community building, through collaboration. But we will also talk about how fermentation can be an expression of social differences, like gender differences, class differences, race, or age differences as well. The next slide. So in the context of a globalized food economy and with this trend of the gourmetization of fermentation that we are seeing here, we have access to all kinds of fermented products from around the world now. So what happens to local food traditions? Are they disappearing or are they just changing? And how are we going to face all this? And also, are we doing cultural appropriation when we make products that belong to other cultural traditions? So in this course, we will discuss how fermentation relates to identities and to heritage, and how these notions of heritage and also terroir, can be used for building political ideas, usually nationalist political ideas. Next slide. And talking about politics, we will also cover how fermentation can be used as a political tool, both top-down and bottom-up. So for instance, we will see how fermentation has been a tool of colonialist efforts to erase particular cultures, for example, but we will also see how fermentation can be used as a political tool for food activism. In fact, I've been working for years with food activists that use fermentation to build mutual aid communities, to build resilience strategies in the context of the ecological crisis, to fight against food waste, also to stand against mainstream food industry, and also to build like healthy relationships with other species. And by the way, you can see in the pictures, well, there's only one picture, but you can see there's like a gender bias in food activism. We'll talk about this in the master. So as I said before, fermentation is a great example of the relationships between humans and other species, because, as Cipriano said, when we ferment, we are cooking together with microbes and enzymes. So it's a collaborative, symbiotic process between humans and non-humans. But this idea of collaboration, maybe it's a little bit idealistic, because in the real world, humans actually are managing microbial life through different strategies and techniques like pasteurization, vaccination, sanitation strategies, fermentation, or, I don't know, genetically engineered microorganisms. So in this context, we're going to reflect together about what kind of ecological relationships we want to establish with microbes, with other species, with our environment, and also with other humans. And how are we going to do that through food production and consumption? And also, how are we going to sustain that? So, next slide, please. In the master, we will cover many other issues that I don't have time now to explain, but one of them is like the artistic practice that use the metaphorical force of fermentation to create around issues such as transformation, decay and putrefaction, social change, purity and contamination, non-human temporalities, the dichotomies of human thinking, the cycles of life and death. Well, there are many things that I don't have like the time to explain now. But next slide, please. As you can see, sociology and anthropology's food is about thinking critically about society and culture. It's about taking into account not only the gastronomic, technological, or microbial elements of food production, but also about the social, cultural, economical, political, ideological, and ecological elements that converge in food fermentation. So with this vision in mind, students will be able to apply all this knowledge to the projects. So this master will give them access to work in, I don't know, in academic research, for example, as a food consultant for both private and public policies departments, for example. They can also work in product design or in hospitality. But the thing is that in these projects, they will be able to like have a critical and sustainable perspective of food systems and manage the projects accordingly. And next slide. They also will be able to conduct social-cultural diagnosis of particular food contexts and make culturally adequate proposals, both for products, for menus, for recipes, for business, or for projects. Understand the consumer trends by understanding food symbolism, food meanings, and the uses of food in each cultural context. And also they will be able to meet the demands of a globalized food market, as well as local economies and traditional practices. So next slide, please. Well, thanks for listening, and I hope to see you in the master's degree. So that's all for me.
We will see you, Arantza. Very interesting. I take this is fermentation go very far, the politics up to the philosophy. So it will be very interesting. Thank you so much, Arantza, to be with us. I'm going to jump to the next speaker. Is Francisco Kuhar. Is a biologist, I will say better mycologist, specialized in biotechnology and diversity. He having working as a researcher many years in CONICET, in the Multidisciplinary Institute of Vegetal Biology in Córdoba, Argentina. Correct me if I say wrong, working with ectomycorrhizas and other different fungi. He also has been curator of cryptogamas in the herbarium of Córdoba, and then they have been doing a lot of stays, I say, for example, in Harvard. And actually, with this long, very long academic career, now Francisco is going to be also professor in the master, but mainly he's a chief scientific officer in NOMI and mycelium specialist in Basque Biodesign Center. But I don't say more. I prefer that you tell us what you do there. Francisco.
Hey, thank you so much for having me here and for this chance of sharing what we do. Actually, I've been working with mushrooms my whole career, and it's a pleasure for me to be a part of this initiative I'm trying to share. So the idea is to see what is the role of fungi, especially filamentous fungi in fermentation, because yeasts are also fungi, but I'm specialized in solid state fermentation because I really believe that when you try to ferment solid things, filamentous fungi are the best because of their ability to penetrate, to form the hyphae. These cells, these elongated filamentous cells, are able to penetrate all solid matters in nature, like soil, wood, or whatever. And when you try to ferment liquids, the best options seem to be cellular organisms like yeasts or bacteria, because they can diffuse and there are currents and they mix them with the medium. So every nutrient is available for them to modify. And the most interesting thing about fungi, but also about other microorganisms, is when you see in nature herbivores. We talk about herbivores all the time, animals that feed on plants. And actually, in nature, there are only a few true herbivores. When you talk about herbivores, those animals we see here, they are not herbivores. They eat plants, but when you see the nutritional needs of these species, they need almost the same amino acids, essential amino acids we need. Essential amino acids are a group of molecules needed to synthesize protein that we cannot synthesize ourselves. We need to eat them in our food. So when we eat only plants, we have to be really specific about the combinations to be able to have all the amino acids and other nutrients we need. But in case of those animals we saw, they have strategies like the fermentation. Fermentation is not only made by humans artificially or culturally, but also in the stomachs or many other organisms. And not only in the stomachs, we will see that later. For example, when you see in the stomach, the first stomach of the cow, we see they have this amazing collection of microorganisms. And I want to show you this one in particular, Neocalimastix frontalis. It's a fungus. It's a weird one, but it's a fungus. And he's in charge of synthesizing all those amino acids the cow needs. So when we say an animal is a herbivore, we have to think that this animal is eating the protein that the microbes and the enzymes are synthesizing in their system. There are almost no true herbivores in nature. And actually, I was thinking in the previous talk when Diego was the name, he was talking about the ants. And I come from South America, and especially in South America, we have those leaf-cutting ants that are the biggest herbivores in the world. The biggest not in terms of size, but in terms of tons. They consume so much plant matter per year, tons and tons. And actually, they culture fungi. And this is the dissertation of a student we had back then when I was teaching at the University of Cordoba. And he was describing actually the fungi they culture. Ants cut the leaves, they take them to their nests, and they culture fungi, special fungi that produce those spheres, those balls that you see in the picture, and they are filled with polysaccharides and amino acids. So when you see animals eating plants, you have to think directly on fermentation. And actually, for us, it's really easy to do that, especially using filamentous fungi, because they grow so well. We can extract a piece of tissue of a mushroom species, like we do in our project, and actually culture it. it's not that difficult like animal cells or plant cells. It's so much easier because you only need like a petri dish with a sterilized medium, and that's it. And then you can build a collection because all those species have different abilities, different enzymes. Some of them can degrade fibers, some of them can degrade pectins, some of them can destroy pollutants. Some of the raw materials that we use for food are contaminated with insecticides, pesticides, or other chemicals, and some of those strains can really degrade those products. So they are interesting too for that. And there are many interesting tests that we can see where we can analyze one fungal species and see all the enzymatic activities they can exert on the medium and on the ingredients, actually just by culturing them on different substrates. And then we can analyze, for example, how much protein they release, how much reducing sugar, how much sugar they release from the polysaccharides, if they produce any bioactive compound that help us with our immune system, or also the antioxidant power they have. And this is really easy. This is something we are doing with a PhD student from me, Antonella, from Argentina. And the best thing about solid state fermentation is that you can make your own chambers. It's not, you probably saw already the chambers, the industrial chambers they use, the bioreactors they use in liquid state fermentation. They are so complex and they need the addition of many purified compounds, but also the control of the gases of carbon dioxide temperature and the production of toxins. Solid state fermentation is a much cheaper process, and it's really interesting for that reason, because sometimes when you want to do food, you need a process to make it affordable, because it's not a good idea to create a new process to produce food that is only affordable for some people, because we want to be sustainable. Solid state fermentation is sustainable. It consumes much less water than liquid fermentations, and also can be used to process subproducts of other industries. But if the price is too high, there's no point on researching on that. This is at least how I think. So, and it's interesting to see also the flavors that we can produce in fermentation, because they are related to the flavors of mushrooms. And that's really fascinating for me, because I really like to read about old stories written from ancient philosophies or physicians about mushrooms. And when you see how they describe the flavors, they have the same problems we have today with the fermentation. So we are analyzing also that, how the people perceives the flavors. And for example, this is a truffle we discovered in Uruguay last year, and we published this piece of research showing all the compounds, the volatile compounds, to analyze the aroma. And this is really interesting when you do fermentation too, because this way you can orient your fermentation to special flavors that you need for your product. And of course, we will talk in the courses about profitability, which is interesting because otherwise you cannot take things to the industry. Scaling up, which is also important because sometimes we think very small scale in the lab, but then we want to feed people. We need to multiply the scale. And also about regulation. We have to keep in mind, and especially here in Europe, that all food that we buy is regulated. Of course, there are other ways to acquire food. We can produce it ourselves at home, but when we want to go to the market, we have to think of the regulation, and especially about fermentation. They are really tough, at least here in Europe. So those are the things that interest me the most about fermentation, and I hope we have time in the future to talk in depth about them. Thank you very much.
Thank you, Francisco. Very interesting. I was telling to my coworkers, like, we should put 21 seats in the classes because I'm going to join to all of your classes. Arantza, Francisco. Yes, for sure. Come with it. Thank you so much, Francisco. We have to jump with the next guest, Pablo Sánchez Rey. Pablo Sánchez Rey, he's a biologist specialized in biotechnology. Pablo actually is a PhD student in the program of Gastronomy Science of Basque Culinary Center. Here we have Francisco, the director. I doing a little help between and Pablo also is a co-founder of Inomi and chief of operation officer. So, Pablo, I would like you to join us, or you want to share a presentation or anything, feel free.
Perfect. Yes, give me a second. Okay, well, hi everyone. Thank you very much, Cipriano, for the introduction, and thanks to the Basque Culinary Center and WebTech for the invitation to participate in this webinar. Well, my name is Pablo Sánchez Rey. I have a degree in biological and biotechnology science, and I have been working with fungi for over 10 years. I specialize in filamentous fungi, particularly white rot and brown rot fungi. I work on several projects such as biomaterial and enzymes production, dye degradation, heavy metal reduction, and for the past 5 years, I focus on developing food products and inputs for the food industry at Innomi. I work with fungi composed of filaments called hyphae. Here we have an illustration about that, which form a tissue known as mycelium. And depending on environmental conditions like such as temperature, humidity, or presence of specific molecules called morphogenes, the mycelium reorganize their structures and form the fruiting body, like the mushroom that we can see in the forest. There are many kinds of hyphae. We have storage hyphae that reserve carbohydrate, lipids, protein, skeletal and binding hyphae that add support to the mushroom, swollen hyphae, gelatinous, secretory. There are many kinds. But to summarize, fungi I classify into three main types: monomitic, dimitic, and trimitic. Don't worry about it. I don't want to bother you about that, but it's important to know the structure of the mycelium, because if we went searching for fungi in the forest, we can find a wide variety of texture. For example, Latipodus sulphureus, that is my favorite fungus, has a chicken-like texture. Even in Misiones, Argentina, people make escabeche with this mushroom. In the other hand, we have agrosive that has a flowery aroma and a particular texture too. Here we have the Manciella canari that has a fleshy texture like meat. And at the end, we have, for example, a Ganoderma species that have a rigid wood-like texture. So this is too interesting for us because these textures are only present on the fruiting body and not in the mycelium. So when we talk with Cipriano and Francisco about that, we ask ourselves, how can induce this texture in the mycelium? And what caused mycelium to develop different texture? So this led me to begin my doctoral research in gastronomy science at the BTC, where we evaluate potential morphogens and the effect of substrate particle size on fungal growth in solid state fermentation, of course. And as an example, here, these two graphs showed the differentiation of hyphae in two strains, in Tamarisis and Torulosa. And you can observe that they not respond in the same way to the morphogens. For example, while one strain increase the number of the hyphae forming node, that is a form of differentiation of the mycelium, the other one show a lower degree of this hyphae differentiation. So for us, it's important to understand how the mycelium, works in the presence of different morphogens. And on the other hand, we study the effect of the substrate particle size on the final texture of the mycelium. We know that the particle size reduces the substrate aeration, which could affect the fungus growth. So this is, well, the full trial is extensive, but has a representation. I'm sharing with you a texture profile analyze, a TPA, of two fermented oat samples grown with the same strain and the same fermentation time. The only variable between them is the particle size. The red one is 3 millimeters, and I believe that is green or blue is 1 millimeter. And you can observe a difference in parameters such as hardness, adhesiveness, resilience, chewiness. So here, the only variable that we have is the size, the particle size, and the results are two different. And what we do with this data? Of course, we can perform a principal component analysis to determine which variables have the most significant influence on each samples. In this case, and I'm sorry, it's in Spanish, but in this case, it's the resilience and the chewiness. And with this information, we can run a multivariate analysis comparing each sample set with the commercial products. And this is the most important thing for us. And even predict which variables need to change to reach a specific target. For instance, if we want, I don't know, for example, to replicate the meat burger, we need to must adjust the variable that bring us closer to that product. And with this graph, we can, for example, we can see the combination of strain, particle size, fermentation time, and the exposure of the certain morphogen in sample G generate a texture similar to that found in the vegan burgers, or well, the vegan burgers 1A and 1B. So these analyses were done using only a few of our strain and evaluating a limited number of variables. So imagine the number of models that we could explore using this type of fungi and the solid state fermentation. So that is why the field of fermentation increasing needs professionals capable of evaluating and predicting this kind of outcomes. And if you are interested in microorganism or an experimentation with fermentation, focusing on food, of course, I encourage you to continue an education along this path. So thank you very much for the time, and I'm looking forward to continue to dialogue with you.
Thank you so much, Pablo. Excellent. This is one of the samples that we can do with solid state fermentation. Pablo, we are looking forward to see you teaching in this master. Thank you so much, Pablo. Now I'm going to just present very quick the master of food fermentation. In food fermentation is the only specialized master actually on the world that is having the fermentation within the focus of the gastronomic science. Fermentation, we said, is a biological and creative force with a potential that is infinite. And we are going to study the fermentation from these three focus that we saw in this presentation: the scientific technical, the culinary, and the social science. Master of Food Fermentation is going to be teaching with our philosophy of learning by doing. So we are going to try that you are going to put your hands on in the lab, in the kitchen, to experience the fermentation by yourself. Here I collect a little slides about the organization of this master. From the culinary is around the 20% of the teaching hour that you are going to have. First semester, you are going to study culinary technique and processes, you are going to have the tools to develop the fermented product from a culinary point of view. And in the second semester, we are going to start to design new food products with the tools that you got in the first semester. Same as the sociology, anthropology, and culture. First semester, we will have Arantza teaching the sociology and anthropology as she explaining in her speech. In the second semester, we will explore how diverse is the fermentation and how different cultures and communities have been using these microbes and enzymes around the world. And in the scientific section, we will have in the first semester food microbiology, food enzymology, and also we will have instrumental and sensory analysis. You will have the tools to know very deep how works the authors of the fermentation. And in the second semester, you will be able to start with the instrumental analysis, the food quality and safety, and to start to work with all this data that you are going to get. Here, this is the other subject in the second semester that appear here. Research and innovation in gastronomy is not in any of these because she's studying how we can study all these disciplines together. So it will be in the second semester. I want to be the key to have all the tools that are necessary to start the master, as Pablo is doing now. The master, sorry, the PhD. Here you have the distribution of the 2 semesters that I told to you. And I'll let you know that you will have a final master project that it will be performed a little where you want you to be. We personalize the company or the university that you would like to be to develop your final master project. What kind of tools we are using? We are using a show cooking and our show labs, formulation classes. We are going to have within this master 3 labs, one physical chemistry, another microbiology, and one lab for fermentation, just for fermentation. We will be placed in the new building of GOE in the city of Donosti. We will have a core project that it will be within the 2 semesters. And we will have one or a couple research and development challenge projects. And also we will try to invite a lot of guests from academia, from companies, to have a real idea about how is the situation about it. And now I can tell you, we just closed it. We are going to have a learning journey in November to assist to the Congress of Science and Cooking World Congress. We will attend and participate in the Congress, and we will visit different food industries, restaurants, and research and development departments within the area of Barcelona. For example, we will be assisting the day of the fermentation. We have here a Pia Sorensen from Harvard is going to be speaking. Just a little brief about the professional profiles. What kind of professional are you going to be when you finish the master? So we can say that you are going to be a food product developer, that you could to develop your activity within the industry or hospitality with the prototypation and the scaling up that Francisco said, or within the academia with the research. So deeply, you will be a gastronomy specialized in fermentation science with strong knowledge of food biotech, but also with all this culinary and social science knowledge. I think we are going a little out of time. Here you can get a little more information about the master, and just open the session for questions and or any doubts that you could have.
Thank you. Cipriano, we haven't got more time.
Also,
With your words and these last comments, we have to close to today's sessions. Those questions that we haven't answered will be responded individually. Yes? And if you have any question, the World Culinary Center team is here to help you. So, moreover, we will send the recording of this webinar by email. So, thank you to our master's degree coordinator, Cipriano Carrero, and our guest panelists for participating today in this webinar. Thank you and see you soon.